Hey everyone! In the last few videos you learned about aneuploidy. In this and the next videos, we'll examine polyploidy, which is when a cell has an extra set or sets of chromosomes. In other words, polyploidy is the presence of 3 or more chromosome sets. It's relevant in humans as an estimated 10% of all spontaneous abortions are caused by polyploidy, which in animals is almost always lethal. However, polyploidy is viable and common in plants, with 1/4 of all species thought to be polyploid, so we'll mostly discuss polyploidy in plants. Now, for this discussion, it's important to remember that one chromosome set has one of each chromosome type. So this is one chromosome set, which again contains one copy of each chromosome type that the organism has. For this video, our 2 questions are: what are the basic types of polyploidy and how do they differ? And how are the genomes of polyploids described? There are a few different ways that different types of polyploids are categorized. One is by how many sets of chromosomes they have. If an organism has 3 sets of chromosomes, in other words, 3 of each chromosome type, it's called a triploid. 4 sets is a tetraploid, 5 sets is pentaploid, 6 sets is hexaploid, 7 sets is heptaploid, and 8 sets is octoploid. Another way to categorize polyploids is by whether they contain chromosomes from one species or from multiple species. Allopolyploidy is when a polyploid has chromosomes from multiple species. For example, consider an allotetraploid. As we saw, tetraploid means there are 4 chromosome sets. The prefix allo means other or different. In this case, it means that some of these 4 chromosome sets are from different species. So here are the chromosomes of a hypothetical allotetraploid. The blue chromosomes are those that came from one species, and the orange chromosomes came from a different species. In this example, both these species were diploid, and through hybridization, they interbred and both passed on 2 sets of chromosomes. The 2 sets of orange chromosomes in the allotetraploid came from species 1, and the 2 sets of the blue chromosomes in the allotetraploid came from species 2. Hybridization can actually give rise to a new species in this way, if the polyploid becomes different enough from the original species and is fertile. Note that I've made the color scheme for chromosomes a bit different than what I've done before. Previously, different color chromosomes were different chromosome types. But for these videos on polyploidy, chromosomes are going to be different colors if they come from different species. Here, orange chromosomes come from species 1 and blue chromosomes from species 2. I'll distinguish different chromosome types by having them be different sizes. So here we see a large chromosome type, a medium chromosome type, and a small chromosome type. An autopolyploid, auto meaning self or same, is a polyploid with chromosomes from just one species. For example, an autotetraploid has 4 chromosome sets, all from the same species. Here, are the chromosomes of a hypothetical autotetraploid, all of which came from individuals of the same species. How we describe polyploids is a little different than how we describe diploids. For both diploids and polyploids, we'll describe their chromosomes both as pictures and as numbers. We'll compare the notation we use to describe their ploidy and how their genomes are represented using letters. Here are chromosomes from a diploid. Here are chromosomes from an autotetraploid. And here are chromosomes from an allotetraploid. When describing the genome of a diploid, the notation is, for this cell, 2n equals 6, where this 2 tells us that there are two sets of chromosomes, this set and this set. And this 6 tells us that there are 6 total chromosomes in this cell. N, as we've defined it before, is the number of chromosome types. Here, there are 3 types: a large type, a medium type, and a small type. So n equals 3. However, n can also be defined as the number of chromosomes present in gametes of that organism. Generally, gametes have half the number of chromosomes as the organism. So each of this organism's gametes would have 3 chromosomes. In a diploid, the number of chromosome types, 3, and the number of chromosomes a gamete has, 3, are the same, which is why n can represent both of these things. But when describing polyploids, such as this autotetraploid, we cannot use n to represent both the number of chromosome types and the number of chromosomes gametes have because they are different. Let's see why. How many chromosomes would a gamete of this organism have? As we said, gametes have half the number of chromosomes that the organism has. This one has 12. So this organism's gametes would have 6 chromosomes, 2 of each type. The number of chromosome types is still 3. Notice that the number of chromosomes a gamete gets, 6, and the number of chromosome types, 3, are not the same. So we can't use n for both as we did for diploid organisms. We need 2 different letters to represent each of these things. In polyploid organisms, n is the number of chromosomes in a gamete, and x is the number of chromosome types. This organism has 2n chromosomes, or twice as many chromosomes as their gametes have. We can also say that this organism has 4x chromosomes, because a tetraploid organism has 4 of each chromosome type. Here, both 4x and 2n equal 12. This is how we write the ploidy of a polyploid organism. 2n equals the ploidy of the organism times x equals the total chromosome number of that organism. Here, the number before x is 4, since this organism is tetraploid and has 4 chromosome sets. Now, if you feel you didn't quite get that, that's okay, but watch the last minute or so again. It's important for understanding the rest of the video. Before we move on, a quick question. How many chromosome sets would be in a gamete of this autotetraploid? I'll give you a moment to pause the video. Okay, so this autotetraploid has 4 sets of chromosomes. Here is one of those sets. This organism will pass on half of its chromosomes, or in other words, half of its chromosome sets, to its gametes. Since it has 4 sets, it will pass on 2 of them. So the gametes will have 2 sets of chromosomes and will actually be diploid gametes. For this alloploid, the notation would be exactly the same as the autotetraploid, assuming we're considering the large orange chromosomes and the large blue chromosomes to be the same type, which we are because we're calling both the large blue chromosomes and the large orange chromosomes chromosome 1s. So 2n equals 4x equals 12. Here, try to answer a second question. What is n for this allotetraploid? I'll give you a moment to pause the video. Okay, recall that n is the number of chromosomes present in gametes, which get half the chromosomes of the organism. This organism has 12, so their gametes would have 6, so n equals 6. Next, we'll list the chromosomes present in each of these organisms. As you've seen with human chromosomes, each chromosome type is given a number. So this diploid has 2 chromosome 1s, 2 chromosome 2s, and 2 chromosome 3s. This autotetraploid has 4 of each chromosome type. So it has 4 chromosome 1s, 4 chromosome 2s, and 4 chromosome 3s. Same thing for the allotetraploid. Now, some textbooks use capital letters to represent chromosomes, but this is not usually how actual chromosomes are named. Plus, I think it's confusing because, as you'll see, genomes are also represented using capital letters. So we'll stick with numbers. Like we said, the genome of an organism is written using capital letters. Usually these are italicized, but I can't really write in italics, so I'll just underline them here. Each chromosome set is given a capital letter, like F. A diploid organism has 2 chromosome sets, so it has 2 capital letters. Again, each F represents 1 chromosome set. In our autotetraploid, there are 4 sets of chromosomes, so 4 Fs, each of which represents 1 chromosome set. Each set is represented by the same letter, F, because they're all from the same species. In the allotetraploid, there are also 4 sets of chromosomes. 2 sets, the F sets, came from one species, while the G sets came from another species. So sets of chromosomes from different species are represented by different capital letters and are called subgenomes. The F subgenome came from one species, while the G subgenome came from another species. Lastly, sometimes you might see chromosomes from different subgenomes actually have different numbers, like this. For example, instead of the 2 chromosomes from each species all being called chromosome 1s, the chromosomes from one species would be called chromosome 1s, and the chromosomes from the other species would be called chromosome 2s. Both of these ways of numbering chromosomes are correct, and different systems are used just depending on the species. Okay, that's all for this video on polyploidy. In the next video, we'll apply what we've covered in this video to some actual polyploid species. See you all then.